Hello, welcome to another screencast where we'll continue our discussion of the immune system. So this is immune system 2, and in this one we'll be talking about describing and explaining the cell-mediated responses that involve helper T cells, cytotoxic T cells, antigen-presenting cells, the major histocompatibility complex, as well as memory T cells and memory cytotoxic T cells, and in memory helper T cells as well. So that's a lot, so let's get started. So in the previous screencast I've shown you this is the roadmap that we'll be using here for the next this screencast as well as the next one, and this is for screencast 2 here. We're going to be talking about this right side here, the cell-mediated immune response. So what we're going to have here is the first exposure to the antigen or to the pathogen. Right? And so the pathogen, again, may be a virus. Pathogens may be a virus, maybe a bacteria, maybe a fungus, maybe a cancer cell, maybe a parasite of some sort. Right? But upon first exposure, the phagocytes, right, that are in the lymph nodes, right? And so phagocytes are cells that phago or eat or consume these things. So you'll have these phagocytes, you have these macrophages here that will actually engulf it, right, by endocytosis and take it in. And when they take it in, they then become an antigen presenting cell, right? So that's what APC stands for. So antigen presenting cell. So these antigen presenting cells will then stimulate here a helper T cell, right? And so these are abbreviated there, T cell and helper, so helper T cells there. And so when you stimulate or activate the helper T cell, the helper T cell will then respond in some way, right, by this arrow here, making more helper T cells, but then also by making these things that are referred to as memory helper T cells. And so these memory helper T cells kind of hang out there a little bit. Those helper T cells, once activated, also will stimulate these cytotoxic T cells. Right? So there's a lot going on here, and it's your job to kind of master the steps involved here. So take a minute now maybe and summarize this pathway here, and then when you're ready, come back. So this is the cell-mediated immune response. We are mediating this by having these cells involved with this. So another thing that will happen in addition to this central pathway here is this pathogen on first exposure, or this antigen here. These antigens are displayed by infected cells, right? So let's say that the virus, the bacteria, and the fungal cell escapes this, or bypasses these macrophages here, and actually goes and infects a cell. Well, these infected cells actually then will present antigen as well, right? And say, as kind of a flag, saying like, hey, I'm infected over here, you need to get rid of me. And so that is going to activate, it says right here, activate cytotoxic T cells. Cytotoxic, cell toxic, cell harming, cell damaging T cells. So these cytotoxic T cells are T sub C cells. These activated cytotoxic T cells then give rise to, that is, they will undergo mitosis very rapidly. Gives rise to, produces here memory T cells, as well as then active cytotoxic T cells. Now these active cytotoxic T cells do this down here. Here's the response of these active cytotoxic T cells. They defend against intracellular pathogens and cancer cells by binding to and lysing the infected cancer cells or infected cells. So again, here is an important theme, right? We have this binding, and when you bind, then you get a response, and the response in this case is lysing of the infected cells or cancer cells. And again, this lysing is to basically cut open, break down, any sort of break down the cells. Okay, So that is the cell-mediated response upon first exposure. Might be a good idea here to pause that before we move on. So let's just review that again here. Upon first exposure, here is your macrophage cell. That macrophage is going to engulf the antigen or the pathogen, and as it does so, it sort of breaks that pathogen down, right? And these antigen fragments then, and so these red triangles are from the pathogen. It might be part of the virus, part of the bacterium, part of the fungus, part of the cancer cell. And so what happens is this macrophage will not only break that down into these little chunks, but it will also produce this thing right here. This protein right here is a class 2 MHC. MHC standing for major histo cell tissue, histocompatibility molecule. It's a protein. Right? It's a type of protein. So genes are stimulated here to then, to be expressed, and you end up making these MHC class 2 proteins. Well, what do these MHC class 2s do? Well, if you look at this diagram here, you take this fragment and you bind it to an MHC class 2, and then that thing actually gets turned from the inside of the cell to the outside of the cell. So now this MHC2 thing, this yellow and red thing, is presenting. So this macrophage is presenting this antigen fragment here. So it's kind of broadcasting to these other cells. It's saying like, hey, look, this is inside of you now, you multicellular organism. You've got to do something about this. So now this cell becomes an APC, an antigen presenting cell. And who is it presenting to? Well, it's presenting to these helper T cells. So here's your helper T cell. And these helper T cells have a T cell receptor on them. Right? And this receptor is, of course, going to now, as you see here, bind with this class 2 MHC molecule. Now, this direct cell-to-cell contact, this direct cell-to-cell binding right here, this binding will stimulate this helper T cell to then have some sort of response. Right? And so this helper T cell will now respond by doing a few things. So let's go from left to right here, and this is, again, going down the center of that roadmap. Here's your antigen preventing cell, this macrophage, here's your bacterium, your pathogen. It's engulfed, and its pieces and parts are broken down. You then have MHC class 2 presenting. Here's your helper T cell. This CD4 and this T cell receptor are binding to the MHC class 2 presenting things there, and this binding right here sets off a whole series of reactions. Now this helper T cell is going to release interleukin and other cytokines. These are chemicals. These are ligands. These are molecules. These are signals. Right? So all these things here, these helper T cells release these things, and the effect is that there is a receptor for these interleukin-2 and other cytokines. Right? Cytokine, cell, kinetic is going to cause cell responses here. So that you have, not shown in this diagram, but you have a receptor here for those on the helper T cells. And what that causes is these helper T cells to undergo mitosis very rapidly, and you end up producing more and more of these helper T cells. The other thing that happens is these interleukin-2 molecules will signal. Again, there's going to be receptors on these cytotoxic T cells, and this is stimulatory. And so the response there, when the interleukins bind to the helper T cells, to the cytotoxic T cells, is that these become activated, and again, cell-mediated immunity attack on infected cells. The other thing that happens is that on B cells, not talked about here in this screencast, but on another one, these will have receptors for interleukin-2 on their surface, and the response there is that you end up getting antibodies produced in the plasma there. Okay, so this is the part of the cell-mediated response. Right? So again, what we just looked at there in detail was this. Right? Here's the cytotoxic T cell, the helper T cell rather. Right? Helper T cells signal to make more helper T cells. You're going to make memory helper T cells, and you're going to make these cytotoxic T cells. Stimulates via the interleukin 2 and cytokines there. Okay, so not pictured here would be another branch off of here where you're going to make memory helper T cells as well. And so another positive reaction there. Okay, so there is that. I think I'll pause here and break this into 2 screencasts. So